Awesome. Well, thank you so much, Dylan. I'm very excited to be there. As Dylan said, I'm with the U.S. Geological Survey, and my name's Elliot Schoenig. A lot of folks are kind of interested to learn how the Geological Survey is working with plants and animals. But there's actually quite a bit of ecology-related work that happens under the USGS umbrella. Back in the day, we used to have a USBS actually. And at one point, when there was some governmental shift going on, that department got lumped in with the GS, and we now have a few different ecology related centers around the country. I'm in the Western Ecological Research Center, which has about 10 or so offices up and down the West Coast, primarily in California. And I'm in an office in Dixon where we have two primary research interests, mostly focused on endangered species with federally mandated monitoring or projects that have to happen. So about half of our office focuses on endangered birds, primarily migratory waterfowl and rails and sage grouse. And then the other half, and this is the crew that I get to hang with, which is much more fun in my opinion, are the folks studying reptiles and amphibians. So we are a herpetology lab at the USGS, and we get to work on a number of really cool species that unfortunately have federal or threatened listing in California or across the whole country. And I pop around to a few different projects throughout the year. But the project that I spend my springs working on and that I have the most fun with, because it's one of the coolest species that we have in this area, is studying San Francisco garter snakes. So I think a lot of folks probably are familiar with this species. It's kind of a very brightly marked snake, and it has a lot of lore around it as one of the most endangered species in our country and globally, and also as one of the most beautiful species. I think it's kind of up for interpretation which snake in the world is the most beautiful, but I think probably everyone would agree that these snakes are going to be up close to the top. They're a really beautiful combo of white and blue and black and red, with these really beautiful turquoise undersides that makes them just a really cool and spectacular species, and just a species that doesn't seem like it should occur in our area. It just seems much more exotic, but it always just kind of gets me excited when I see one just kind of cruising through a grassland or a coastal scrub system, because it just seems so out of place with how bright they are. And the San Francisco garter snake is in itself very endangered, but it's actually a subspecies of a much more widespread species. And so many folks are probably familiar with garter snakes. They're one of the more common and one of the more easy to bump into snakes that people see hiking, especially around the Bay Area where we have quite a few different species. They're prone to kind of moving about in the day when people are active. They forage in the open around water areas. So unlike a lot of our snakes that are very secretive, they tend to be seen quite a bit. And the common garter snake is one of those species. It's one of about 30 species that occurs around North and Central America. And this is the most wide-ranging of those species. And in each kind of part of the country, and I should say the continent, because this snake occurs all the way from Alberta up in Canada, all the way down into northern Chihuahua. I think actually the southernmost populations may be at the southern tip of Florida too, which makes it a little further south there. But yeah, really wide range north to south, and then also east to west across the entire country. And you can see each of these colors on this range map here represent a different subspecies of the common garter snake. It displays quite a bit of variation across its range, and in a number of distinct areas, certain populations have developed really different patterning and often really different ecology too. And so these populations aren't quite different enough to be described as unique species, but they often have some sort of, they may be in the process of evolving from a broader species. Subspecies can be kind of like partly differentiated. They can have really unique genetics or really unique patterning or really unique ecology, such that they warrant kind of recognition and also protection in their own right. And in California, we have a few different subspecies of these common garter snakes. But we're going to be looking right here just around the Bay Area, because the San Francisco garter snake has the smallest range of any of these subspecies that span across the whole continent here. And it just occurs right around mostly San Mateo County and in the Santa Cruz Mountains region, from kind of northern Santa Cruz County now right up to kind of the San Francisco San Mateo County line. And we're going to talk a little bit about IDing San Francisco garter snakes, but I'll just point out too that there's this whole red range around the dark blue here that is the range of the San Francisco garter snake in our region. That is a similar looking garter snake called the California red-sided garter snake. And so often folks may tell me that they saw a San Francisco garter snake up at Point Reyes or maybe down in Monterey here. But there's a similar snake that looks pretty similar that occurs around our area. It just so happens that on the San Francisco Peninsula itself, the snakes evolved some really special patterning. And so that patterning is distinctive in identifying this snake. Primarily, they're going to have a very red or orange head, and then they're going to have a series of lengthwise stripes that run down their body. And the top stripe is going to be some shade of yellowish or whitish, often with kind of a bluish tint. And then that's going to be bordered by black stripes, and then these really unique red or orange lateral stripes that kind of run down the side of the snake. And then usually another black stripe followed by kind of this blue side stripe, which is kind of an extension of this really remarkable pattern that comes up from the underside. They have these really blue ventral patterns or belly patterns. And I mentioned that the snake looks similar to the California red-sided garter snake, which occurs down in Monterey and in the East Bay and in the North Bay. But in the Santa Cruz Mountains proper, the San Francisco garter snake does coexist with two other garter snakes that superficially kind of may look like it. The Santa Cruz garter snake is very different looking. It doesn't have any red patterning and mostly just looks like a highway, I like to say, just like a dark pattern with a big yellow line running right down the center. So that one you're not super likely to mistake, but coast garter snakes are very variable. And they can have extensive red checkering, but they don't have this clean red stripe that the San Francisco garter snake has. They can also end up having a lot of red or orange speckling on their heads, but they're not going to have this entirely red or orange background color to the head. It's usually going to be a little greener. And occasionally, like very rarely, you'll see one that you might describe having like a bluish tint, but it's never going to be really blue like the San Francisco garter snake. And then range, again, is going to be really helpful for identifying this snake versus the California red-sided garter snake. So here's kind of another look at a few of those different pattern characteristics. We've got that really red head on both of these two San Francisco garter snakes. This was a male-female pair that I think was mating in one of our snake traps that we'll talk about in just a minute. And then you can see that after the dorsal stripe that's just barely visible, you get this alternating black, red, black, and then these turquoise blue sides that kind of run down into this really, really blue belly. And that blue belly is just kind of a really unique color for snakes in general. There's not a whole lot of blue snakes, so it's just very spectacular looking. And here's a comparison to some of the garter snakes you might be likely to confuse San Francisco garter snakes with. On the left, we've got a coast garter snake, which we can see has a lot of this red patterning, but the red is kind of more the background color. It's kind of like a checkery background. Also with some kind of speckly red spots that kind of go down onto the underside and the sides. And then it's got a very greenish, yellowish brown head. Very strikingly different from the red head on this guy in the middle. And then on the right is a California red-sided garter snake. Again, that's that subspecies that occurs up against kind of the surrounding parts of the Bay Area. And they can occasionally be even more blue than this pictured individual. They can be just about as blue as a San Francisco garter snake, and they have that reddish head. But you'll notice that the red is distributed along the side of the body in these really clean kind of red or orange squares. And at some point, the snakes in our area, in the Santa Cruz Mountains and along the San Mateo coast, something mutated in their genome, and whatever codes for the red patterning along the side shifted from those red checkers that are common in California red-sided garter snakes, as well as most of the other subspecies, or many of the other subspecies of common garter snake. And it shifted into this really solid, striking red line that we see in the San Francisco garter snake. So again, these are different subspecies, and subspecies in reptiles and amphibians don't coexist. Very occasionally, or in other species, I should say, it occurs more common where two subspecies may meet. The populations can kind of what we call intergrade, which is kind of like a hybrid between subspecies, and you might get individuals that could look similar to both of these two. And there's one population that we think is probably an intergrade population between these two, which is down near Palo Alto. But basically, everywhere else in the Bay Area, you're either going to be looking at just this one or just this one. And range is really gonna help clinch that identification. So these are kind of the two species that you would be most likely to confuse for a San Francisco garter snake, but we actually have really, really good snake diversity along the San Mateo coast and in the Santa Cruz Mountains in general. Sorry, I got a fire truck coming by. So these 8 species here are the most common species that we catch when we're out looking for San Francisco garter snakes. We've got the San Francisco garter snake in the bottom left there. As you can see, it's got that nice red stripe and a really red head. And right above it is another like really spectacularly orangey red coast garter snake. But again, it's got that greenish head instead of that beautiful red head. And the red isn't really formed into that perfect stripe. To the right of that is that highway Santa Cruz garter snake I mentioned earlier. Really can be super striking with this dark yellow stripe alongside it, but doesn't look all that similar to the San Francisco garter snake, aside from the fact that it has a stripe. Underneath that is a little Pacific ringneck snake. These guys are more fossorial, which means they spend most of their time underground, but we see them alongside San Francisco garter snakes quite a bit. And then on the right, we have kind of 4 of our more terrestrial snakes that occur in patches of coastal scrub and clearings along the San Mateo coast. We got a little gopher snake, and then a big rubber boa in the top right. Below that, we have a northern Pacific rattlesnake, the only venomous snake that we have in the Bay Area. And then to the left of that, we have a yellow-bellied racer. So these are the 8 species that when we're out looking for San Francisco garter snakes, we're most likely to encounter. But there's a handful of other species in the Santa Cruz Mountains as well. I think all in total, there's 15 different species or subspecies of snake that inhabit kind of the Santa Cruz Mountains region. So it's a really diverse spot for snakes in general, which is super cool because it makes it a fun place to study them. Because as you'll see when I show you kind of how we catch them, it's kind of like you never quite know what you're going to get. Because anytime you could walk up to a trap and you might have any of these 8 species inside of it. So primarily, San Francisco garter snakes are occurring alongside wetland habitat. They are mostly frog eaters. The large adults generally hunt our California red-legged frogs. And then the smaller juveniles and even mid-size and large-size adults will still eat the California or the Sierra and chorus frog, the little tree frog frogs that are hopping around. And so those two species make up most of the diet of San Francisco garter snakes, and both of those species lay their eggs, and then those eggs hatch into tadpoles in water features. And as a result, San Francisco garter snakes really need a permanent water source to maintain a thriving population, because that's where they're hunting those frogs. They are kind of semi-aquatic, we like to say. They do occasionally hunt in the water proper, but oftentimes when we find them, they're just kind of creeping right along the water's edge, and they're looking for frogs that are sitting or hopping around along there. But upland habitat is also just as important as this wetland habitat. And in the background of this photo, you can see just some really extensive coastal scrub that's kind of extending down into this big lake. And in the foreground, a big open clearing. And we found that this kind of mosaic of scrubby habitat with small openings is kind of the most preferred habitat for the snakes. We found when we did some vegetation analysis compared to the habitats around our trap areas that the most likely traps to catch the snakes were the areas that had the most diverse suite of plant species that occurred around them. So they don't really like a monoculture of any one type. Don't want just grasses or don't want just coyote brush. They want a mix. And usually, like I said, they're close to water, but they can disperse through upland habitat and cross pretty long distances. So occasionally we'll catch snakes at this body of water here. And there's another small pond up and over the backside of this back ridge, and snakes will cruise up through the scrub occasionally and get to those other ponds. And so scrub seems to be a kind of a linker to different ponds. It kind of like bridges the gap between them because it maintains a little bit more humidity. Maybe there's some frogs or salamanders hopping around, and it gives shelter to the snakes. So we've looked at kind of the beauty of the San Francisco garter snakes, but I mentioned kind of two of the things it's most known for as being one of the most beautiful and also one of the most endangered snakes that we have around here. And its decline is kind of has happened for a number of reasons. One of the first is thinking back to that range map that we saw. This was never a species that was widespread or subspecies that was widespread. It always was very restricted to just our region. And so with that said, it was never going to have a huge population because there just wasn't a lot of habitat for it. But what habitat there was, unfortunately, quite a bit of that was lost. And much of that is due to being at the edge of a huge urban area, especially further north, up in Pacifica and north of there. Some of the areas where this snake used to occur are now just neighborhoods in kind of the sprawl of San Francisco. And then we'll see another map in a minute. Some of the kind of more agricultural lands along San Mateo coastline also are no longer suitable habitat just because they were altered too much to provide appropriate foraging and upland habitat for the species. So loss of habitat is a big one. They also suffer from effects of invasive species, and this is a little bit harder to quantify. We don't know exactly how big of an issue this is, but a former coworker of mine, Rich Kim, did a lot of work looking at the direct interactions between bullfrogs and San Francisco garter snakes. And this is a figure from a paper he published about 5 years ago now, based on a bunch of work our lab did. And really what he found to be the biggest issue with bullfrogs was that they were a direct competitor of San Francisco garter snakes, more so than a direct predator. So they're eating the same prey that the garters like, whether that's these Sierra and tree frogs or even mid-sized red-legged frogs. Bullfrogs will kind of eat whatever they can get inside their mouth. He did confirm that bullfrogs did occasionally eat San Francisco garter snakes. So there definitely was also a direct relationship between that effect. Other invasive species that harm this snake include crawfish and bass. Similarly, these like may occasionally prey on San Francisco garter snakes, bass more so. But the big kicker is that they're again competing. Bass are eating the frogs and the tadpoles, and crawfish are really tough because they really hammer the frog egg masses. So again, it's just kind of limiting the amount of prey that is available for the snakes to find. So invasive species is really tough. Another issue that has been kind of exacerbated by all of these other problems is inbreeding. And as the populations have shrunk and declined and become more separated, there's less and less gene flow and connectivity between them. And the result of that is that in some of these areas, snakes have really no way to connect with their closest neighboring populations. And that has caused really disastrous effects at a few different sites. And we're kind of working on some ways to resolve that I'll touch on at the end. And lastly, I'll just mention disease. Similar to invasive species, we don't know a whole lot about how this is affecting San Francisco garter snakes currently, but you may have heard in the last 7 or 8 years or so that what we call snake fungal disease, or ophidiomycosis, has appeared in California. And this is a fungal infection that was really detrimental to some snake populations in the East Coast. And when it kind of first really reared its head, it really hammered some populations of some threatened species over there, primarily some denning rattlesnake species like Massasaugas and timber rattlesnakes. And in certain areas, it almost wiped out entire populations of snakes. In addition to rattlesnakes, it was really bad for garter snakes, king snakes, rat snakes, and racers. And one thing that all of these snakes have in common is they tend to congregate in den areas in the winter. And this is more common on the East Coast than it is in California. Aside from kind of the really high elevation parts of California, snakes don't hibernate in a traditional sense, especially in the Bay Area where we have pretty mild winters. And so many of you may have taken a hike on a nice warm December or January day. And if it's been sunny for a couple days, you're pretty likely to encounter a snake maybe out basking on the side of a trail. But in these parts of the East Coast, like every snake in a whole hillside would come and find one outcrop where they could kind of get below the frost layer. And the result of that was that these dens kind of had the perfect conditions for growing a fungus. They were cool and damp. And then also it was the perfect conditions for spreading that fungus because all of the snakes would come together and kind of cuddle up for the winter. So in California, we have now found ophidiomycosis in a handful of species. Many of these detections, though, are asymptomatic snakes. And so they are carrying the fungal pathogen, but they are showing no signs. And so we don't, for that reason, know if it's having a big effect yet. There have been some snakes, in particular a handful of king snakes that have appeared that do really look pretty rough, kind of similar to this water snake here. But we're still kind of learning about that. It's very new to this whole equation, but it could have played a role in the past, and maybe we didn't even know about it. So all of those, in addition to one of the kind of former threats, which was poaching, many of these snakes were taken from the wild in the past, which is hopefully less so of an issue now, but was certainly a problem. All of those issues have kind of led to a very fragmented population, but one that still occurs across kind of much of the kind of footprint of its historic range. So it still occurs from all the way down in northern Santa Cruz County, up into one or two spots along the crest of the mountains, along Skyline, a few spots on the east slope of the Santa Cruz Mountains, up to around Pacifica. It's just that you might notice a huge gap of spots in the middle here. And this is not truly reflective. So there are a few populations in this area, but there also are a number of areas where snakes have apparently gone extinct in this large central zone. And the same is true further north and on the east slope of the Santa Cruz Mountains, where there's quite a bit of development. And this is what's kind of really impacted this inbreeding issue, because most of these populations, kind of with the exception of this southern coast region here, and then the area around Crystal Springs Reservoir System, they're just too isolated to really have regular contact with other snakes. So that's kind of like a little backstory on the snake and the issues it's faced. What we get to do, the fun part is that we get to kind of go out and look for these snakes and study them in all of these remaining areas to figure out how they're doing. And you guys are very lucky to work in the state park system that has a few populations of this beautiful species. Año Nuevo is one of the most widely kind of known populations, and snakes are pretty prone to just kind of basking on the side of the trail. I would bet a few of you may have encountered them there at some point. But there's also populations at other parks in Pescadero Marsh, and then also at Rancho del Oso, which is Big Basin. They're significantly harder to encounter or detect in both of those areas, potentially because those they kind of are tricky to find in these big estuarian ecosystems, or areas where these creeks and rivers kind of come out and have these big marsh systems right before they enter the ocean. So it's almost like looking for a needle in the haystack. Whereas at Año Nuevo and at other sites where they're kind of around these ponds, they're kind of like, it's like this discrete location that you can kind of sample pretty effectively. And that makes it really hard to know for sure how many of these snakes still may occur at other areas further north, like San Gregorio, Tunitas, and Pomponio, all of which where there's state beaches at the mouths of these creeks. And likely there might not be reproducing populations of the snakes at these sites, but at least every once in a while, snakes may kind of disperse down from maybe larger populations further up in those watersheds. So our work at State Parks Land has primarily happened at Año Nuevo. My time at State Parks with USGS studying San Francisco Garter Snakes started in 2018. We did a pretty big range-wide demographic study looking at kind of population growth, population size, as well as it was kind of combined with a genetic study. We did that in 2018 and 19. And we did a bunch of estimations of population sizes. And one of our sites was kind of around the big pond at the visitor center there. So we spent a lot of time there. And that was, coupled with a genetic study that kind of was resulted in our discovery that a lot of these populations are kind of inbred. But Año Nuevo, because it's kind of this big connected ecosystem of ponds, from there basically up to Pescadero Marsh was one of the least inbred locations. So really promising. More recently, we have done a little bit of distribution work and habitat assessment in some of the less studied areas of the park. There's a few kind of more outlier ponds that we occasionally go visit to try and confirm ongoing presence. But what's occupied most of our time recently has been a response to restoration work that happened at one specific parcel of Año Nuevo. And I'm going to kind of outline that. But first, I'll just kind of show you how we're catching snakes. This is kind of what I mentioned earlier that makes this such a fun area to study snakes in, because what we get to do is we set out traps. And these traps are set right along the side of a short little drift fence. And when the snake is cruising along, it hits this drift fence, and then it's hopefully going to turn left or right. And then whichever direction it goes, there's going to be a little funnel trap right at the end. And so on the right side here, you can see this is a rubber boa who's followed the fence line. And it's just entering the funnel here where it's going to drop into the trap. And these funnel traps are kind of like a crab trap or a lobster trap where the snake could find that same hole to get back out, and certainly they occasionally do, but for the most part they just they just are content to hang out. And I always like to kind of show this comparison because some days trapping is really awesome. I think when we're like studying a rare species, this is what our expectations or our dreams are going to be. That we're going to come back and we're just going to be traps are just going to be loaded with them. Um, but the reality is we're catching a lot of the more common species, coast garter snakes, or these are racers on the right that were mating inside of a trap. We sometimes see these big balls of coast garter snakes when a big, large female gets into the trap and a bunch of males will follow her in. So this is what we see more often. And actually, I should just have an empty trap because that's what we see the most often, or maybe a vole or a mouse. But this is the snake wise what we see. This is what we're hoping for. And you'll probably also notice, I'm sure I'll get a question at the end. I'll just answer now. These sponges that we put inside the traps, we keep these really saturated and wet, and they help keep any amphibians, especially red-legged frogs that might wander into the traps. Just kind of gives them a good wet source of moisture to hang out in the trap so they don't get too dry. The other thing that we do to catch these snakes is we set out cover boards. I have a little video here. Let me just double check that. Okay, yeah. I have this little video here. So what we do is we many folks use wood cover boards and lay them out so that the snakes can kind of shelter and thermoregulate, kind of warm up underneath them. We found that in these coastal regions, the thing that works best is actually corrugated roofing metal because it warms up really nicely in the late afternoon. And so we put these out around some of the ponds that we work at, and we come back. And hopefully it'll look something like this, where we got a mating pair of San Francisco garter snakes that's hanging out underneath. And you'll notice when I put this piece of tin back down, this side right where the snakes were is kind of right in the sun here. And so that's kind of where they were hanging out and just kind of staying warm. And I had happened to see those same snakes the day prior, so I let them do their business and hang out. But normally we're going to be capturing them to mark them because that's how we really get an idea of how many snakes there are at each of these sites. And so we have a little hot cautery unit. It's a little, we call it a micro brander. And we essentially give each snake a little brand. And we do so, you can look in the bottom left corner here. These are the snake's kind of ventral scales. They're kind of like a big segment that moves up their body. And we start at this, what we call the vent or the cloaca, and we count the scales going up from there. And doing that, we can kind of give each snake a unique code. So on one side, we count up and we go 1, 2, that's a 2-digit. Then across the body, we go 1, 2, 3, 4, that's a 40-digit. And then after you get up to 90, we jump to the hundreds. And so this is a 500-digit year. And so all that adds up to 542. And this unit is really hot. And snake scales are keratin. So I like to show folks in person that I can kind of brand the back of my thumbnail. And if I brand the thumbnail, I don't want to go through it because then I hit my flesh underneath, which hurts. And similarly, you can brand through the scale and you don't want to hit the flesh underneath. But if you just do this outer scale, this racer here has really fresh brands that kind of singe up. It doesn't harm the snake, and then it lasts for the rest of their life. This is another garter snake here that's scarred up in this really black pattern here. And it makes it really easy to identify the snake upon further collection. And we do that so that we can know if each snake is new, which helps us when we're doing analysis to estimate how big the populations are, how many of the snakes are surviving year to year, seeing if snakes are moving around individual snakes. In addition to marking, we also do some morphological data collection. So we measure the snakes along a ruler here, and then we also weigh them. This is my field partner, Patrick, who's done a lot of the field work out here with us. And this info helps with our growth analysis, and also it can be a backup tool if we're unsure on identification of a brand. You can check to see, is it a boy or a girl? Is it big or small? The last time it was caught, and that might limit the options that you have. So we're going to talk a little bit about this Green Oaks project, and this is a site where we have employed a lot of these same techniques. So this is a parcel in the northern, or I guess north of the visitor center at Año Nuevo, and this was acquired as part of a mitigation project where BART was encroaching on some San Francisco garter snake and red-legged frog habitat, and they bought this site that was a farm and donated it to the state park system. But it had San Francisco garter snakes and it had red-legged frogs, but the vast majority of this whole area I've got squared in here, and even parts outside of that, was just kind of weedy grassland, and it was not providing good habitat for either species. It had one really big existing pond that was the irrigation pond, and then that drained through this really channelized ditch through the parcel to this wetland system in the bottom. And there were garter snakes at this pond and this wetland, but in many, many years of coverboard surveys and trapping, we never encountered them anywhere outside of these two water features. And so after kind of really confirming that they were not utilizing this habitat, just about 5 years ago now, State Parks partnered with San Mateo RCD, and they worked on a project to significantly alter the site. I kind of brightened up this next image just to try and make it a little more apparent, but I also put squares around what happened, which was a returned the site to its more natural hydrology, which was to say they filled in this really channelized ditch that cut through the site, and pushed kind of the draining groundwater into this swale system. And along the swale, they added 5 new ponds, and then they also added a 6th pond kind of along this different swale. And then they heavily altered the existing ag pond to kind of make it a little more natural. So we've got all of a sudden this burst of new habitat. And in 2023, we started a 5-year monitoring project to determine how snakes were doing. And in 2023, we caught snakes at this pond and this pond, which is the first detections anywhere in this central part of the parcel. The next year, we got snakes at this pond. The following year, we got a snake here. And just earlier this spring, we caught snakes at this pond and this pond. And we still have been encountering snakes around the ag pond the whole time. So at this point now, we're only about 4 years post groundwork, and we've already got snakes utilizing all of the new pond features that were created. So a really awesome success story. And because we marked all these snakes, we're able to kind of estimate the population size in this area. And it gives us this nice, clean increase in snakes each year, which I think as a landowner or a land manager like State Parks is, or landowner and manager, this is really what you guys want to see. You did this restoration work, and we started out, there were not that many snakes. Probably many were kind of pushed out of the area temporarily during all the groundwork around that pond. So the first year we only estimated there were about 10 to 12 snakes. The next year, about 20 to 25. Last year, about 35. And this year, we've already encountered more snakes than we did last year. Likely going to be a higher estimate again. So really awesome. Range wide, we've worked on a little bit of other projects. At one site, this is not at State Parks land, but nearby, we have a 20-year monitoring project going on. And that has been really awesome because we're still kind of learning quite a bit about natural history of this snake. Quite a bit is still unknown, even though it's so like protected and beautiful. It's not all that studied. And so this was a snake at that Green Oaks site that regurgitated a live leech. And that was a new prey item that they had never been recorded taking before. So even after a 20-year monitoring project kind of across the whole range and focus in one area nearby, we had never seen that before. And so we're still learning quite a bit of new stuff. That said, we're going back to most of these sites about every 5 years. That'll kind of be our plan once we wrap up this monitoring project at Green Oaks. That's what's mandated in most of the Endangered Species Act related legislature. And like I said, we're kind of doing these genetic health assessments that have really illuminated that one reason some of these populations might be struggling is because they're really inbred. And that has kind of led to what I'm going to talk about for the last couple minutes here, or last bit of this, which is that we're finally kind of really getting to a big conservation action with this species. We've been really trying to learn as much as we can about their natural history, about their population growth and movement, so that we can really make sure we get this right. But that's all leading us to starting what we call a head starting project. And head starting projects, I'll kind of give you a little backstory on. I think a lot of folks think about these big conservation actions as being kind of like captive breeding programs, but usually before captive breeding is tried or warranted, we'll start with a slightly softer effort, which is to try head starting. And what that means is that we are raising the juveniles, the babies, in captivity, and then releasing them back into the wild. But we're not actually breeding the adult snakes in captivity. And I'll explain a little bit more about how that all happens. But the reason that this is really important is because when these snakes are born, they are tiny. They're only about 2 grams. They're only about 7 inches long. And they are food for everything. I mean, even like red-legged frogs, bullfrogs, definitely, other snakes, birds, everything would eat one of these things in a heartbeat. And in addition to that, their small size just kind of limits their ability to really be active. Snakes kind of in general are thought of as loving warm and hot weather, but they're also a body type that really heats up and cools down quickly because they're cold-blooded. So they need to be able to warm up or cool down to maintain what they want their body temperature to be. And as a result, this tiny little, basically like thin little piece of spaghetti in a hot sunny day heats up super, super quickly, especially more so than a bigger snake that has more what we call like thermal inertia. And once it's kind of at its certain temp, it'll stay there for a little bit longer. So that small size also just limits the amount of activity these guys can have. And that all leads to what we call really high mortality in their first couple years of life. And so this graph here is a what we call a survival graph. On the x-axis is the length of the snake. So this is in millimeters. And this is the body length of the snake. So very occasionally we get snakes that are body length around 80 centimeters or 800 millimeters, right around a meter total with their tail. But when they're born, they're really, really tiny. Like I said, just like 7 or 8 inches, right around 200 millimeters total. And so the annual survival, which is on the y-axis here, is significantly lower for these snakes during this first stint of their life. And really, you can see this part here, it's rapidly increasing as their length increases until it gets to kind of a point in the middle where it plateaus and it's pretty level for a while once they reach their adult size. And so head starting is a way to keep these snakes in captivity and kind of supercharge their growth through this first period, such that when we release them back into the wild for the first time, because they're born in captivity to wild moms, they kind of avoid this first period where they're really, really prone to getting eaten. And this means that basically we need to have a really good idea of which mother snakes are pregnant or gravid, what we say, out in the field, and how many babies are they each going to have, especially based on size. Because again, we've done all this monitoring. We really want to make sure we know as much as possible. And that's kind of led to our use of this portable ultrasound device that we carry out into the field with us. Um, and I have this photo on the left here, which shows kind of the length of the snake. So you can see at the bottom here, this segment that kind of looks like bone is a vertebrae as the snake is moving down. So this is basically like the snake's head is in one direction and the tail is in the other. And as we're moving along, you see each of these three little eggs here or embryos developing. And this is what it looks like to ultrasound a snake out in the field. This is my partner and I. And I've also got a little video kind of moving along the length of the snake. And so you can see each of these little kind of discrete packages is like its own little embryo. And yeah, so this allows us to get really good data on how many babies each of these snakes in the wild is going to have. And that's led to us kind of getting these really good models that allow us to predict the likelihood of reproducing for different moms based on different size, which, as you can see here, the gravid moms are these dots on top. Moms that are not gravid are the dots on the bottom. And this is the probability of a snake being gravid or reproducing. So being pregnant, I should say. It's a more common term. And as they get bigger, they're much more likely to reproduce. We're much more likely to see these embryos along their body. Additionally, as they get bigger, we're more likely to see more embryos. And so this is really kind of showing us that for this head starting project, the bigger the snake, the more babies they're going to have. And sorry, 1 second, I'm just going to plug my charger in. Okay. And so that is been really critical information for us to learn, because this is all allowing us to predict the. Oops, sorry about that. I'm in my field vehicle right now. I'm in between field sites, so bear with me for the this. Okay. And so this has been really important for allowing us to really know which of these snakes is going to have the most babies, and which of them should we bring into the wild, or sorry, bring out of the wild and into captivity to start this head starting. And in addition, we did a ton of simulation work to really make sure that we knew what the best option was. So we simulated moving adults from one site to the other. We simulated bringing them into the wild, or bringing them out of the wild, and releasing subadults, releasing babies, and then also the length of reintroduction. So how many subsequent years we're going to do this. And all that to say that we really are taking our time again to make sure that we're really getting this right, because we don't have too many options at this point, and we also don't really want to get it wrong, because the effects could be disastrous. This is the likelihood that we might cause extinction of the donor population where the snakes are coming from, which is also really important to consider. So we really crossed our T's and dotted our I's, did as much work as possible. And the results is that we now have baby snakes being born at the San Francisco Zoo. This was a super big milestone last year, where we brought pregnant moms into the zoo finally. And those babies are now about a year old, and they're getting pretty close to the size at which we're going to release them into the wild. And so that is kind of our next big step in San Francisco garter snake conservation, is that we're going to start moving these snakes around, both to kind of help populations that are low on numbers, but really to kind of try and get this refreshed genetic diversity that could be so critical for kind of keeping some of these sites from blinking out, because there's only so much reproduction that can go on with the same snakes again and again. At a certain point, they just can't reproduce anymore. They fertilize the eggs. So that's like a really promising step that we're at right now. We've got all these babies. They're hopefully going to get released later this year at a number of sites. And the cool thing, going back to this, head starting thing, is in a normal wild clutch of snakes, we might see that only this is like an optimistic guess that maybe about 30% of snakes at this really small size here, 40% of snakes are going to make it to their first year of life. This is that survival. But with head starting, because we have basically the entire clutch of snakes that is surviving in captivity, we can actually release about 30 or 40% of that group of babies back where the mom would have been given birth. And we can essentially simulate what would have been a natural recruitment. And again, that goes into this simulation work that we've done. And if it all works out like we really have been focused on making sure we think it will, we'll essentially be able to kind of pinch off some snakes that theoretically wouldn't have survived in the wild at the site they would have been born at, and kind of replenish the genetic diversity and the numbers at some of these other sites that really need it. So that's kind of leading to future work at Año Nuevo. I mentioned we have another year of post-restoration monitoring that we're going to be doing to hopefully see yet another bump in snakes that are occurring at this restoration area. And then we're going to be kind of back to a 5-year or approximate return schedule, where we're coming to make sure that they're stable. We're hoping to work and do some abundance studies in other parts of the park. I mentioned there's some areas where we occasionally go to confirm, see that snakes still exist. This is the case too at Rancho del Oso. There hasn't really been much work to determine how many snakes are at these sites. We just occasionally get confirmation that they're still around. But that would really be helpful to kind of know how many populations actually exist on state parks land and how big they all are. And then similarly, hope to get to visit some of these sites further north up the coast between Pescadero and Half Moon Bay, some of these estuary areas that have historic records, but where nobody's encountered them in quite a while. And range wide, we're going to kind of continue these genetic health assessments to kind of figure out how successful this head starting has been, especially once we hopefully have a couple generations of breeding back in the wild. We'll be able to see if the genes from the population where the snakes came from are now showing up in the wild snakes where they're released, which will be a sign that it's working. An additional part of that is that if this all goes well, we're hoping to start moving snakes to sites where we know they are extinct. So this would kind of be the next big step to kind of reestablish populations that have been lost. And hopefully Año Nuevo might get included in some of that work because, like I mentioned earlier, it's one of the sites that has the highest genetic diversity and would be a really good site to include in some of this work. So that's what we have kind of moving forward. And I got to just shout out all of our partners at all of the field sites that we work at, whether that's Mid-Peninsula Open or Regional Open Space, Peninsula Open Space Trust or Post. Obviously, you guys at state parks, we're so grateful to be working here. And then our regulatory agencies, CDFW and U.S. Fish and Wildlife, as well as San Mateo RCD, who's doing so much good work out here, and many, many more folks too, that are really integral with everything that's going on. And with that, I think that's all I've got for you. So I think I'll open it up to questions, and I think I will end my